Imagine, just for a second, a blue whale. Just picture the sheer, unbelievable scale of it.
Oh, yeah, the size is just hard to even wrap your head around.
Right. And it is swimming through this freezing ocean, totally, perfectly engineered for the water. It's got those massive flukes, a blowhole, the thick insulating blubber.
Exactly. It's an oceanic survival machine.
Yeah. Now, put that image right next to a pigeon soaring over a city skyline. You are looking at hollow bones, feathers, a beak, a body that is completely optimized for the sky.
Totally different hardware.
Exactly. And finally, put yourself in that lineup.
Yeah.
A human being walking upright on two legs, holding a smartphone, navigating the land.
So you're looking at three organisms with entirely different sets of survival hardware, basically. I mean, they could not be more radically different in how they look, how they move, and where they actually thrive.
Yeah, we look like we belong to completely different universes. But here's the massive puzzle we are diving into today. Okay, let's unpack this. If you were to wind the clock all the way back, long before the whale was born, before the bird hatched, and before you were even a thought.
Back to the very, very beginning.
Right. If you look at the very early developmental stages of these three exact creatures, that radical difference completely vanishes. It strips away all our, mammalian bias.
It's wild. It is honestly one of the most profound visual tricks in biology because the adult forms scream diversity, right?
Yeah.
But when you look at those early embryos...
And just to be clear, an embryo is the organism in its early developmental stages, right?
Yep.
Starting from just a single fertilized cell that starts dividing into specialized tissues.
Exactly. When they are just that tiny organizing cluster of cells beginning to differentiate, you see a totally shared blueprint.
Which brings us to the core mission of our deep dive today. We've got this really fascinating stack of research on embryological development, and we are using it to answer one driving question, which is, what can embryos reveal that adult bodies hide?
Because there's a whole secret history written into us before we even take our first breath.
It's wild. Like if you look at a turtle, a pig, and a human at roughly, say, day 15 of development, they all have a prominent postanal tail and these weird little grooves in their neck. I see these unlabeled embryo matrices in the sources, and it is, well, it's almost impossible to tell them apart.
It really is. Even trained biologists can struggle if you strip away the labels. You don't see a tiny turtle with a little shell or a miniature pig with a snout.
Right, there's no tiny pig nose.
Exactly. You see this distinct, curled, almost sea-like shape across the board. You see little blocks of tissue called somites that will eventually become vertebrae. The visual similarity is just staggering.
But looking at this data, it forces a very specific kind of thinking. Like it feels like you can't just jump to conclusions. You have to actively separate what you are physically seeing from what you are deducing.
Yes. That distinction is the absolute bedrock of scientific literacy. When scientists look at that unlabeled grid, they have to start with the strict observation, the raw data.
Just what's right in front of them.
Exactly. The observation is simply, I see a prominent tail and a series of folds in the neck area on all these early embryos.
No claims attached to that. It's like looking at the construction sites of different buildings. At the very beginning, a skyscraper and a house might just look like identical concrete foundations.
That is a great way to put it.
You only see the differences as they get built up. Just the facts in the petri dish. But then comes the leap of logic, right? The data doesn't come with an instruction manual, so scientists have to infer what those similarities actually mean.
Right, the inference is the profound part. Scientists observe those shared macroscopic structures and conclude, well, they are not just a random coincidence.
Like, what are the odds?
Exactly. The only logical explanation for a human, a pig, and a turtle starting with the exact same complex physical features is that they inherited that genetic blueprint from a common ancestor population that lived millions of years ago.
I buy that we share a blueprint. I really do. But because these early embryos look so strikingly similar in that grid, I keep getting stuck on how easy it is to oversimplify this.
Oh, absolutely.
Like, are they exact copies of each other? Because I remember seeing old biology textbooks where the embryos of different animals basically look like absolute clones in the first stage.
Yeah, you are likely thinking of the legacy of Ernst Haeckel.
Haeckel, right.
He was a prominent 19th century zoologist who took a perfectly valid observation of similarity and stretched it into a deeply flawed inference. Haeckel heavily promoted a rule he called ontogeny recapitulates phylogeny.
Can we get a translation on that?
Sure, yeah. He argued that as an embryo grows, it literally replays its entire evolutionary history.
Literally?
Literally. He claimed a human embryo physically becomes a microscopic adult protozoan, then morphs into a microscopic adult fish,
Mhm.
then an adult reptile, and finally a human.
Like hitting a fast forward button on millions of years of evolution in just a few weeks. I mean, that sounds incredibly elegant, but biology is rarely that neat.
Yeah, biology was wonderfully messy.
Yeah.
To make his theory look flawless, Haeckel took some, let's say, heavy artistic liberties with his illustrations.
He fudged the drawings.
He really did. He actively omitted structures that didn't fit his narrative, like the massive yolk sacs attached to some embryos, and he artificially resized his drawings so the early stages of different species looked perfectly identical.
Oh, wow. So he smoothed out the messy reality to sell the neat theory.
Pretty much. But modern high-resolution photography has completely dismantled Haeckel's specific claim. Embryos of different species are undeniably similar, which provides brilliant evidence of common ancestry, but they are absolutely not identical. We don't climb an evolutionary ladder in the womb. We radiate outward from a common starting point. A human embryo is never a miniature adult fish.
Okay, well, here's where it gets really interesting, because you just brought up the fish thing, and I hear this myth everywhere.
Oh, I bet.
People confidently claim on the internet that human embryos have fish gills. If we never go through a literal fish stage, what exactly are those matching grooves in the neck that we observed in the Matrix?
Yeah, debunking that fish gills myth is crucial. Those little folds in the early human embryo are scientifically known as pharyngeal arches.
Pharyngeal arches.
Right, they are a perfect example of homologous structures, meaning they share a deep structural and genetic origin across all vertebrates. Now, if you are a fish embryo, as your development continues, those arches will eventually differentiate into functional gills to extract oxygen from water.
Okay, but I'm stuck on the logistics here. If the shared DNA says build a pharyngeal arch, how does the body suddenly know halfway through development to pivot?
That's the big question.
Like, we obviously aren't breathing underwater in the womb. Is there some sort of master switch that flips to turn that tissue into something else?
It actually comes down to regulatory genes, specifically a group known as Hox genes.
Hox genes.
Yeah, HOX. Think of them as the foreman on a biological construction site. They don't actually build the bone or the tissue themselves. They control the timing. They dictate when other genes turn on and off.
Oh, I see. So the base blueprint is the same, but these Hox genes change the work schedule.
Exactly. In mammals and birds, those exact same starting arches are heavily modified by these genetic switches. The cells migrate. Instead of growing outward into gill filaments, the tissue folds inward.
Then what does it become?
It differentiates to form the specialized bones of your inner ear, the malleus, inbuccusis, and stapes, as well as your lower jaw and the cartilage in your throat.
That is wild. We don't have fish gills. We have the starting materials a fish uses to make gills, but our genetic foremen direct those materials up into our skulls to build our hearing and chewing hardware.
Perfectly said. The fact that your ear bones and a fish's gills start from the exact same microscopic folds of embryonic tissue is incredibly powerful evidence that humans and fish share a distant evolutionary ancestor.
But if we start with this ancient shared foundation, how do the final results end up so drastically different? Like, a whale's flipper in a human hand start from the same little limb bud in the embryo. How does the process actually diverge so radically?
Well, to understand that divergence, we have to look at evolutionary developmental biology. It's often just called Evo-Devo.
Evo-Devo.
Kitschy.
It is. And it revolves around what happens when populations get separated over deep time. We are talking hundreds of thousands or millions of years.
So like a single group of animals being split by a river changing course or a mountain range rising up, and they wind up in totally different habitats.
Exactly. Once separated, natural selection begins favoring different traits. Yeah. But here's the thing. Evolution rarely invents totally new macroscopic structures from scratch. It modifies what is already there.
It works with what it has. Right. Think of the ancestral genome less like a rigid blueprint and more like an old family recipe book.
Okay, I'm tracking. Like a recipe for a basic cake.
Right. So evolution doesn't erase the old recipe. It just crosses out bake for 30 minutes and writes bake for 45, or scribbles a note in the margin to add chocolate chips.
Ah, right.
The embryo still starts by mixing the same basic flour and eggs. Those are those ancient homologous structures we saw in the Matrix. But the later genetic instructions heavily modify the final result to fit the new environment.
So the environment acts almost like an editor. Like a whale embryo and a human embryo mix the exact same batter for a limb bud. But the whale's genetic recipe says, keep growing the tissue between the fingers to make a giant paddle.
Precisely.
Meanwhile, the human recipe says, trigger programs cell death so the tissue between the fingers dissolves, leaving us with separate digits to grasp tools.
Yes. What's fascinating here is how often we see these genetic margins scribbled full of edits in ways that are just startling.
Like what?
Take baleen whales. As adults, they have those massive comb-like baleen plates in their mouths to filter krill out of the ocean. They don't have teeth at all.
Let me guess, the embryo tells a totally different story.
It really does. Deep in the womb, embryonic baleen whales actually develop tiny tooth buds.
No way.
Yeah, the ancient ancestral recipe for teeth is still executing. But before the whale is born, a later genetic edit kicks in. The tooth buds are completely reabsorbed by the body, and the baleen begins to grow instead.
Wow. So the embryo physically manifests the evolutionary history. It literally starts building the teeth of its land-dwelling ancestor before the new instructions kick in and say, never mind, we're doing baleen now.
That's exactly what happens, and we see a similar phenomenon in certain species of snakes. They briefly develop tiny leg buds as embryos.
Snake leg buds.
Yep. The ancient genetic sequence for limbs starts to run, and then the regulatory switches halt the growth. The limb buds vanish long before the snake hatches.
That is just incredible. But if our embryos are essentially holding on to this ancient genetic baggage, I'd imagine we'd see similar evolutionary leftovers in the adult bodies too, right? Or does all of it get perfectly erased and hidden by the time we are fully grown?
Oh, the adult bodies definitely retain the evidence, which is crucial for building a solid scientific consensus. Because in science, you never want to hang your entire theory on just one hook.
Right.
You need backup.
Exactly. Embryology provides a powerful, undeniable window into the past, but it is constantly cross-referenced with comparative anatomy in fully formed adults.
So looking at the actual skeletal structures under the skin.
Yeah. Take the forelimbs we were just discussing.
Yeah.
A bat swing, a whale's front flipper, and your arm.
Mhm.
Externally, they are totally customized for flying, swimming, and grasping. But if you take an X-ray of all three, you find the exact same homologous pattern hiding inside.
This is that classic one bone, two bones, many bones layout, isn't it?
You've got it. A single upper arm bone, the humerus, two lower arm bones, the radius and ulna, a cluster of wrist bones, and finally the digits. Fascinating.
The recipe book was heavily edited by natural selection to change the lengths, thicknesses, and fusions of those bones for different environments, but that underlying structural arrangement is unmistakably inherited from a common ancestor.
It's like finding the exact same architectural framing inside a modern skyscraper, a suburban house, and a suspension bridge.
That's a great analogy, and if we connect this to the bigger picture, you also layer the fossil record on top of this anatomy.
Right, digging up the past.
Exactly. The sedimentary rock acts as Earth's chronological diary, showing these physical, structural changes progressing from simpler forms to more complex ones over millions of years. And the final, most definitive line of evidence is modern genetics.
Actually reading the recipe book itself.
Sequencing the DNA of these organisms reveals the exact same patterns of relatedness. When you have multiple independent lines of evidence, fossils in the rock, the one bone, two bones anatomy in the adult skeletons, the DNA sequencing in the cells, and the temporary structures in the embryos,
They all line up.
all pointing in the exact same direction. The scientific argument for evolutionary relationships becomes incredibly robust.
It's a web of evidence that is just impossible to ignore. It all ties back together perfectly. So what does this all mean? We started with the sheer, baffling diversity of a blue whale, a pigeon, and a human. Adults that look like they belong on different planets.
Very different planets.
Right. But our driving question was, what do embryos reveal that adult bodies hide? And the answer is that embryos reveal the hidden, temporary structures of our shared evolutionary past. They are tiny, living archives of history.
They really are.
They show us the rough draft, a blueprint that our adult bodies have heavily modified, edited, and sometimes completely hidden away just to survive in our current environments.
It completely changes how you look at the natural world. Every living thing is carrying its history with it.
The next time you get a chance, jump online and examine some embryo images for yourself. Look at the early stages of a chicken, a pig, and a human. But don't just look at them as weird little pictures.
Right. Treat them as raw scientific data.
Practice your observation skills and ask yourself which of these physical similarities are meaningful evidence of an evolutionary relationship.
And, this raises an important question, and one that I think is totally fascinating to ponder. We've talked about how the folds in a human embryo's neck are the exact same starting structures that become fish gills, But in us, they are reprogrammed by Hox genes to become our jaw and ear bones.
Right.
Since we know these edits happen, what other invisible vestigial remnants might be briefly appearing and disappearing inside us before we are even born?
Wow.
What other tiny, temporary structures are acting as a physical timeline of ancient genes entirely hidden from the outside world?
It really makes you look at a pigeon on the street or a picture of a whale and realize that deep down, at the very beginning of the biological construction site, we all started with the exact same foundation. Thanks for taking the deep dive with us.